Yeah, so Brad couldn't be here today, so he sent me to introduce Becky. But Brad met Becky at a short course doing Bayesian statistics in Colorado, and immediately I think was enamored with your statistical skill and conned her into coming multiple times to come visit us and work with the students in our lab. But Dr. Tang received her PhD in statistical science from Duke University, where she worked with modeling applications in environmental and ecological science. Becky currently works as an assistant professor of statistics at Middlebury College, where her work includes developing joint species distribution models for various plants and animals. She's brought her statistical expertise to eBird data, fossil records, forests. I think I've convinced her to start thinking a little bit about fish also. But today she'll be talking to us about mechanistic modeling of climate effects on redistribution and population growth, which I will now let you take away.
Thank you. All right, can everyone hear me all right? Okay, I'm just gonna be walking with this mic. I don't have something to clip it on. Great, well, thank you for the really lovely introduction. I'm so happy to be here again at Stony Brook. I am going to be talking about what you see here, which is a bunch of words. Hopefully will make a little bit more sense by the end of the talk. But this is joint work that I did with my PhD advisors, Jim Clark and Alan Gelfand at Duke University, along with a student who's now a postdoc at UNC, a marine ecologist, Sarah Roberts. And this is work that was recently published in Global Change Biology, which is very exciting for us. Okay, so I am a Bayesian statistician. For those of you who don't know, maybe you'll get to see some Bayesian stats as you progress through your graduate career. I'll talk a little bit about that. But as a statistician in general, one of our goals, a lot of what we do is we do a lot of modeling. And so I think there's a general understanding of what a statistical model is, but if you ask someone to give you a definition, I think it's very hard. And so when I talk about statistical modeling, I mean, you guys probably have fit linear regression models or logistic regression models, just regression models in general, but really it's about trying to come up using mathematical expressions. You're trying to define the way that you believe the data are generated under certain statistical assumptions. That's all it is. And so if you think about linear regression, you think that your predictors are linearly related to the outcome response. But once we're talking about modeling, we have a bunch of different goals. And this is sort of my overarching, when I try and build a model, this is what I think about. So what I want to do is I want to learn about unknown quantities in my model. And I will call these parameters. So those might be like the regression coefficient, the slope set, if you're used to linear regression. And I also want to, so I want to learn about their values given the data, but I also, it's not just about what I know, but how well I know it. So there's a lot of uncertainty quantification that we care about as statisticians. So that's talking about confidence intervals or credible intervals, right? We also really want our models to be interpretable, especially when we're working with ecologists or other scientists in other disciplines, right? We want to make a model that they can understand and they can put in their expertise, right? So I'm trying to leverage expert information. I collaborate a lot with ecologists. They know way more about, you all know way more about ecology than I do. And so I want to use your years of experience to help me build a model that makes sense. And so this comes in for me as a Bayesian, where we say the experts usually have some prior beliefs about the true values of these parameters. So this might be the prior. I might believe that it ranges, the value ranges from 0 to one side of the axis, and the text is probably small. But I might believe, this is sort of how I believe the parameter behaves, its distribution. But then I want to update these beliefs in the face of data that I've collected. You do all this field work to collect data, to test your hypotheses. Once I have that data, I should update my beliefs, my prior beliefs, after observing the data, and that leads to something called a posterior distribution. So you're going to see these words prior and posterior in my talk. Really, it's prior is what I think, how I think these parameters are going to behave before I see data, and then once I see data, the data and the prior come together to make a posterior, right? It's combining both what I know or what you know as an expert with the actual data. Okay, so Talking a little bit more about, I did not write these obviously, but people have done a lot of research, talking specifically now towards the application of that's coming at the end of the talk. People are interested in climate change and their effects on different animal and plant communities, right? So people are understanding that temperatures are changing and that's causing fish to move, that's having some impact, probably a negative impact on fish and fisheries. And they're trying to understand where are fish moving in the face of climate change, for example. Where the populations, are they staying where they are? Are they dying out? Are they going to places they weren't going before? So they're trying to understand, a lot of scientists are trying to understand how climate change is a real issue. What are the impacts for the populations of interest? And so there's this good paper in Science that's talking about improving the forecast for biodiversity under climate change. And what I love about this paper is what I've highlighted here. It says most current predictive models exclude important biological mechanisms, such as demography, dispersal, as evolution, and species interaction. So as the statisticians, we can make models as whatever models we'd like. Right, but they often exclude really important behaviors that you ecologists know govern how populations behave. Right, we have our math, we have mathematical assumptions, but we want they're missing the ecological assumptions. Right, and so because of that, current modeling efforts are probably not providing sufficient predictions, which is really important when we're trying to understand what kind of policy should we be making to impact or protect future populations. So that's just the graphic of some examples in this paper. These are like 6 key biological mechanisms that we know exist in nature by studying these populations. We know that species interact, we know that species move, but they're not usually getting built into models. And so my advisors were really upset with that. And so we talk about species distribution models in the statistics world and ecology. Everyone's all about SDMs, JSDMs. I don't know if you've heard that term before. But species distribution models are models that try and understand or trying to shed light on the abundance of species in relation to their environment. And so from a statistical point of view, they're often correlative in the sense that we have some statistical distribution that we place on the data and we relate it environmental covariates, but they're really the statistical assumptions. We say that there is perhaps some correlations between species and a community through residual covariance, but we don't necessarily have evidence of that in the data itself. So on the other hand, there's the mechanistic side of things, which is where we're trying to incorporate those biological processes that I just presented on the previous slide that we know or suspect are operating with the community. So is there a way that we can actually embed the notion of species interactions into our model as opposed to saying, oh, there's just some correlation between species? Is there a way that we can actually directly put it in there? And we hope, the point of the mechanistic model is that we hope that we can better represent the system of interest. Okay, so the research goal for this talk, for the work that motivated this talk, was we're trying to develop a dynamic model, so dynamic here meaning time series, a dynamic model that is both mechanistic and probabilistic in order to better understand how the biological processes and how are operating with the community and how the species in the community are reacting to environmental change. Big, lofty goal. And so we're just taking one stab at it. And so once again, to reiterate, mechanistic here is we're going to be specifically incorporating some biological processes into our model. We can't cover all of them. There's so many out there, but we're just taking a step forward. So in particular, what we embed into our model are species-species interactions, so the notion of community dynamics, as well as the environmental response and redistribution or movement within a region. And what we say by probabilistic is that we assume that our data are observed with error. So some randomness going on. There's a random process as opposed to sort of a lot of mechanistic models assume are deterministic. They assume no error at all. The species will redistribute exactly according to this, or it'll evolve exactly according to this equation. We say, no, there's going to be some error in there. It's going to propagate through our model. Okay, so a lot of those words, hopefully these next things will It's not too statistical. I'm really trying to break it down in terms of the ecology that I had to learn a little bit about. But we're going to talk about sort of the way that the data are formatted for this talk. So we have a predefined region, which is a big blue square, that has also been predefined or broken up into a bunch of different grid cells. They don't need to be equal sized, but just for the sake of me making a PowerPoint slide, it was very easy to make them have equal area. All right, so we have n different aerial units, and I'm going to refer to those with the index I. So if n of these here, n would be 16. All right, so we have one big region with n aerial units, and within each aerial unit, we have data on the abundance of capital J different species. Like here, J would be 3. All right, so I have the measured abundance, for example, of fish. You could have birds per, I don't know, some point radius, for example. And then, so that's sort of like a one year snapshot. But we have multiple time points. We have capital T time points, which I will refer to as years, but they could be months, days, etc. And we're going to assume equal time points, equal speed. All right, so we've got multiple, so we've got a lot of data on hand, right? We've got multiple years, multiple aerial units, and multiple species. All right, so, and the shading here is meant to show sort of like there's probably some environmental gradient in this region of interest, and probably multiple gradients depending on the predictors of interest that might influence the behaviors, the growth behaviors of these species. Okay. So here is a high-level view of the model in pictures. I'm going to step through each part one by one, and I'm going to show very little statistics. Hopefully, you will be able to all follow along. There will be a little bit of equations, but hopefully you'll have seen some of it before. So what we're trying to do in our model is we're trying to create a model that we hope can represent or capture the true process. Now, once again, we know that there's so much going on in this system of species of fish species, this community that's just that we can't capture. It would be sort of impossible to do that, but we're trying to, go one step forward and saying it's not just probabilistic. We're trying to put in these biological mechanisms. You're kind of hopefully notice that there's sort of 2 layers to this graphic, where in reality the data that we observe most likely are not exactly equal to the true process of interest. Right, we take noisy measurements of data, or there's some latent level. So we measure something, but what we really care about is some other underlying factor. And so this bottom row here is meant to is the latent process. So what we don't really get to observe, but we hope that we are gonna. That's how we're assuming that the populations are evolving. And then the upper level is what we get to see. So those are the observations that we get to take. So we're trying to use the observations that we see to learn about the latent process, right, where we embed those biological mechanisms. So species interactions don't happen just on the fish that we observe, they happen at the latent level of all the fish. So, in particular, growth and movement are going to operate on the latent scale. And so we're going to start, and this is extending the work of one of my advisors, Jim Clark. So we're going to start with the growth process. It's probably my favorite part to talk about, and hopefully it feels a little bit familiar. So we are going to assume in our model that local population growth, so by local, I mean within a specific aerial unit. So we're looking at one of those little 16 squares. Within a specific aerial unit, we believe that population growth is influenced by species-species interactions, which are denoted by these little arrows back and forth. Environmental species interactions, so the black lines with that thermometer, as well as some independent growth. So species are just going to grow because they have some density independent growth rate. Right, so we believe that these are the mechanisms that are going on that are influencing the increase or decreases of abundances in that aerial unit. And after all of these, of course, this is a constant, these are constant processes, but for the purposes of a model, we're going to assume that they kind of all happen at one go. And after all of these interactions take place, the abundances, the true abundances within that aerial unit are going to change. Right, so now you'll notice that the size of the fish in these going from one point to the next point have changed. All right, so that's pictorially what's happening. The question is, how do we embed that mathematically or statistically into our model? So this is where the ecology comes in with Lotka-Volterra equations. Who's heard of Lotka-Volterra? Oh, okay, great. Well, for those of you who don't, no worries. I had never really heard of it before I started these projects. So we're going to take it from a broad view. We're not going to get into the math too much. But Lotka-Volterra equations are a set of differential equations so we're going to consider sort of two populations, and then we can generalize this. But let's assume that we have two species, species 1 and species 2, right here, so denoted by that subscript. And W1 of t is going to represent the abundance, the true abundance of species 1 at time t. And similarly, W sub T2 of t is going to represent the abundance of species 2 at time t. And we're going to assume that these two species interact with each other. So maybe some kind of bird, some kind of worm, right? Or a shark with a kind of fish. And so these are sets of Lotka-Volterra equations are sets of differential equations that are defining the instantaneous growth rate. So these are sort of in a very, very short window of time, how much are these abundances changing for each species, right? And so what we're going to do is we're going to try and make equations for these instantaneous growth rates that build in those mechanisms that we just presented, right? That's going to be, this is how we define how growth behaves. So, let's make sure that's okay. Great. So, in a Lotka-Volterra, so this is nothing new, this is very old. In a Lotka-Volterra model, we assume that each species has its own density-independent growth rate, which describes sort of how much it grows irrespective of any outside influence, right? So we know that rabbits breed like crazy. Their instantaneous growth rate is going to be probably very large compared to large predators, which have a very small instantaneous growth rate, right? They don't have that many babies. And so when we talk about building out the instantaneous growth rate for the first species, for species 1, we are going to take its current, dropping the parentheses t for notation, we're going to take its current abundance at time t and just multiply it by whatever that independent growth rate is, that density independent growth rate. So it's modulating. Sort of, we're taking the abundance now, and we're saying it's going to change according to, by this much, according to its density independent growth rate. Then we're going to allow for species to interact with each other. All right, and so this is called interspecific growth for us. And so we might say that, okay, species, I know that I have two species, a fox and a rabbit, and here fox eats rabbit, so that's providing energy for the fox. It's also putting a negative influence on the rabbit population. All right, so that's sort of interspecific competition. And that's going to get built into the model with this term right here. I'm going to call it alpha sub 1, 2. But how much the growth of this first species changes is going to depend on both where that first species, how much of the first species we have, as well as how much of the second species we have, right? Because of that competition component, right? It depends. If there's a lot of predators out there, that should be factored into how much that first species abundance changes, right? And it gets modulated by this alpha term right here, alpha sub 1, 2. And then a species may interact with itself, and this is called intraspecific competition. And so this might be sort of like, as we'll see later on, like I've been told that fish don't really discriminate. They'll eat whatever swims into their mouth, including species of their own, they're the same species for some fish. So that would be some sort of an intraspecific competition, or maybe they're competing for space. And so that gets built into this term, this last term right here. And so you'll notice that we have a squared term. It's just basically the same idea as the second term here, but now we're just taking 2 copies of the abundance. Right, and so that gets modulated by the intraspecific growth rate, which is at alpha sub 1, 1 right there. And so this is sort of, so the way that we read these subscripts, in case it's helpful, is that it's talking about the effect of the second subscript on the growth rate of the first subscript. So alpha 2, 1 is saying the effect of species 2 on species 1, because that's the interspecific competition right there. Okay, and interactions can take on many forms. You can have competition, you can have predator-prey, mutualism, but that's sort of what I mean by interactions right here. And so the signs of those alphas, whether they're positive or negative, might tell us about the kinds of interaction. So if species 2 is a predator for species 1, then alpha should have a negative sign, that we would expect it to lead to a decrease in the abundance of species. Right? If there's a lot of if the predators are having a predation effect, we expect a decrease due to that predation. So the signs will be important for us. Oops. And then what we do, just a little bit more of this is going to be a little helpful later on. What we do is we essentially just collect all of those alphas into one big matrix to have them all in one place, essentially. So I had an alpha 1, 1, and I had an alpha 1, 2, but you can imagine that we also needed to create that differential equation for the second species as well. So there's going to be like an alpha 2, 1 and an alpha 2, 2 as well. And we're just putting them all into one big matrix right here. Right. And some things to note about this matrix is that it doesn't need to be symmetric. So when you think about predator-prey relations, alpha 2, 1 and alpha 1, 2 are going to have opposite signs. The predator is going to have a negative effect on the prey, but the prey will have a positive effect on the predator. So we expect those signs to be different. But also, this matrix, some of these coefficients, these alphas, don't need to exist. They can be 0, right? Maybe you believe that there's actually no direct pairwise interaction, so you can just put a 0 in for those alphas right there. So in this sort of 3 by 3 matrix, we would be saying that species 1 and species 3 don't interact. All right, so each one of these alphas are going to be a parameter in our model. So if you learn about law coupled terra in like an ecology class or differential equations class, these growth rate terms are taken as known. But for us, we're trying to estimate these alpha terms. So we're going to put priors on them. We're going to estimate them in our model. All right, let's see. Then the second biological mechanism that we're trying to put into the growth rate is this notion of environmental species interactions, which is the notion that the abundances of the species depend on the environment in a non-linear way. So it's not just like for every 1 unit increase of temperature, it's going to lead to exactly this 1 unit increase in abundance. And so we might have P different predictors, and they enter in with, they get treated sort of very similarly to the previous slide, where if we're looking at the first environmental predictor, and its effect on the first species is just going to be entered through X1, W1, so it gets multiplied together. So this is what I had on the previous slide. Right, and then we just add in all of these environmental species interaction effects. So it's if I'm focusing on species 1, and it's just we have species 1 all the way through, and then we're multiplying the actual covariate value, the predictor value for those predictors, and they get their effects get modulated by these growth, which are showing the effect of the ESI environmental species interactions growth. Hopefully I haven't lost too many people. All this is saying is that we're trying to put in community dynamics and also environmental species interactions into this model. And so those row terms are also other parameters that we're trying to estimate. Okay, so this is probably the most mathy slide, but it's not even really that much math. I'm just going to now generalize this to capital J species. So on the previous slide, I had 2 species. But in reality, in a community, there's probably more than 2, right? And so for capital J species, all we really do is we just add in all the different interactions between the specific species and all the remaining capital J minus 1. So we just build these in. So all of this is the same as before. I'm just adding in indexing to denote aerial unit and general species J. And then this second line here is just showing all of those community dynamics right here. And so what we do just to shorthand it is we put these into vectors to make the math look a little bit nicer later, but that's just for us. It doesn't take away from the idea here. Okay. So what we would do in a local terra model is you would say, okay, so then my abundance for the J species in aerial unit I at the next time step, so T plus 1. So if I'm at T now, the abundance in the next time step would be where I am at the current time step, plus all of this growth stuff that we just talked about. Right? So it's where we are now, plus the changes according to the growth model. That would put us at their abundance for the next time step. This right now is deterministic, right? This is saying how much I expect to see at time t+1 is exactly equal to this equation, which in theory is probably not true, right? Our animals do not behave exactly according to this model right here, to this equation. So I'm going to make this stochastic. I'm going to put in some uncertainty. I'm going to allow for some variability. So I'm going to call this, just for notation, I'm going to call this whole thing on the right-hand side m sub ijt. And so I'm going to say, well, instead of having exact equals, I'm going to say, well, actually, I believe that the abundances at the next time step for species J in unit I is going to be normally distributed, but centered at this growth sort of equation, but it's going to have some error. It's going to allow there's some uncertainty there. So it doesn't have to follow that exact equation, right? Allowing for, this is giving in some flexibility and probably better represents the true process. Right, there's definitely more going on into this growth process that I've captured right here. And so I'm just making a little note for my just for to be true to the model. Instead of having it be at time t+1, I'm not moving. I'm going to just the timing, the notation gets a little bit crazy because we have two processes going on, and it's not already crazy enough. We have two processes going on. We have growth, and eventually there's going to be redistribution. And I don't want it to be, oh, growth takes us to t+1, time t+1, and then redistribution takes us to time t+2. In reality, those processes should be happening in the same year, right? And so for notation purposes, I'm saying, okay, the growth process leads to changes in the abundance, not for the next time step, but I'm acknowledging that there's some change. So that's going to be just denoted with the W tildes. These W tildes are the post-growth abundances, essentially. But if you want to think of it as next time step, totally fine. Okay. Great. So now we've done the growth process. Or we've written down, believe me, that's sort of how it works in our model. And now we're going to allow for redistribution. So now that these communities have grown, now they're going to move around the map. All right. And the question is, how do they move around the map? And there's no good answer. If we knew, that would be great. But they don't really respect these aerial units that humans have put on to this map, right? And so, but the idea is we're trying to do our best to capture some notion of the redistribution process. We're doing our best here. And so the idea here is that species or different amounts of populations can either stay within their current aerial unit, or they can feel free to move out of their aerial unit into other ones, right? And so they can go sort of, some can stay and some can move. And so we're going to try and do, we're going to try and build that in. We're going to try and mathematically write down a way that allows for this sort of redistribution behavior. And the redistribution is what's going to take us to time t+1. Okay, so you know there's t and then t+1 right here. How these species move, how they disperse from one aerial unit I to another aerial unit I prime is well studied depending on each species, right? Like people spend years they collect data, they try and understand how these animals move around the map. And so there are many ways that you can think about it. You might believe that there's sort of a constant rate of dispersal. So if this is our map and we start in the center square, maybe it's realistic to believe that within a given time, there's equal sort of chance of moving from this square to any other square in this region. I don't know if that's like, you could probably think of a species or a system where that makes sense, but most likely there's going to be some notion of distance involved in redistribution. So most likely species are more likely to redistribute or disperse to areas closer to them than further away, right? And so that's the idea here. So there are some dispersal mechanisms, or some people have put into models the idea of a neighbor-based dispersal or redistribution, where you're only allowed to move to one of your neighboring regions, so a bordered one, and everywhere else is sort of zero chance. And then we can relax that assumption a little bit by having it truly be distance-based. So the population can move anywhere in the map, but most likely they're going to have higher chance of staying close to home than they are going far away, right? And so this could be extremely species specific. If you think about, I don't know, sharks are probably more likely to be able to swim to faraway regions than tiny little fish. They don't have as much power to them. And so we would like in our model to allow for redistribution to vary by species. And we also, these are sort of fixed notions, but we also like to bring in some unknowns into here. I don't believe that this is exactly how these species redistribute. So how do I build in a little bit of flexibility into here? I think last slide with math could be it. So this is where we are on the previous slide. So we have this post-growth abundances that I just talked about. So after interactions, after ESI, we've got these abundances. I'm collecting all for species J, I'm collecting them all into one big vector. And then all I'm doing is I'm saying I'm going to define a matrix H that exactly determines, that tells me the probability or the chance of moving from one unit to another unit in the map. That's all this H is. It's saying, here's how I believe these species are moving and redistributing across the map. And so we're getting the abundances, the latent abundances at time t+1. We're saying that that's equal to where they were post-growth at time t after redistribution. Right. And there's just a little bit of important things to keep in mind, that we need to preserve total abundance. We don't want to allow abundances to randomly increase or decrease for a given time year, but that's not accounted for in our model. So we need to normalize these columns. But all this is to say is that if there were 14, if the abundance is 14 for this given species at time t, we want to make sure that after redistribution there is still 14 the next time. We don't want to have lost any. Okay. So these right here in this H matrix, the values here are predefined, but we actually don't know those. So what we might want to do is have them have a functional form, depending on some parameters that bring in sort of this notion of maybe temperatures are impacting how or where species are moving. So we're going to bring that in later on as well. But it really determines on the system, right? I can't say that there's going to be this redistribution matrix for all species. It really depends on if you're working with insects or fish or birds. Okay, so once we've redistributed, now we get to the observation part, which is the only data that we really get to see. So someone goes out and collects some fish, and the observations are taken up here. So once we redistributed, we observe at time t+1. And so, as we can probably guess, right, the bottom layer was the true latent local abundances. But if someone caught 0 of this third species of fish, that probably doesn't mean that there's 0 of that fish existing in the aerial unit. There's some observation error going on, right? Maybe it was too small, or maybe it was misidentified or something. Right, so what we want to make sure to do is say that, okay, the observed abundances are noisy measurements of these true latent ones. So we're going to take our data, we're going to use these data, which we refer to with Y's, and we're going to use that to try and learn about these W's and learn about those alpha parameters and those rho parameters. Right, that's all we get. All we have are these noisy observations, which are probably not the same as the true abundances. And so what we do is we link them together with a statistical model, a log normal model, because you will probably have crazy abundances, but this is just a way to link the observed data to this true latent process that we've been building, that we've built to the biological mechanisms for. And so there's some error as well. Okay. It's a lot of me talking. I guess that's the point of this. But so that's sort of the broad level model. I'm not going to really talk too much else. Hopefully you get some idea of what we're trying to do. Pictures are coming very soon. But this model, as I coded it up, is extremely complex with lots of parameters to estimate. I mean, good thing we have lots of data. The more data you have, the better you can estimate parameters and increase number. But we try and do our best to make our models a little bit more parsimonious, or try and restrict our model a little bit to have sensible inference in that sense. So what we'd like to do is we'd like to leverage prior information or expert information into our model to sort of so we can have more targeted inference. If I know that this is the behavior, if I know that these two species don't interact, I should build that into my model. I should not have to estimate that interaction parameter. I should just go ahead and explicitly set that to 0, and therefore give more data to estimate the other parameters. And so I have these pictures. How do we fit this model? I'm a Bayesian, so Bayesian modeling actually allows us to take this really complex process and break it down into sort of little building block pieces. And so the abundance of time t, if I know the previous time point, I don't care about all the other time points, essentially. So this is saying there's sort of conditional independence going on. But as a Bayesian, this model's actually, I mean, it is terrible to fit and to code up, but it could be a lot worse. So, oops. So in terms of, I don't expect everyone to know these words, but for those who do, so like I said, you can sort of a priori, I mean, just like ahead of modeling time, before even seeing data really, you can set some of those interaction coefficients to 0. So if I know that these two species do not interact because they live at completely directly interact, because they live at different trophic levels or something, then I'm going to set those to 0. And then what we can do is we can say also, maybe I'll go to this one right here. Maybe I know the nature of the interaction. Maybe I know that these two are predator-prey relationships. So I'm going to set the priors, what I believe the behavior of those coefficients to be, as either being entirely positive or only allowed to be negative. That way, I can target my inference to leverage the fact that people have done a lot of research about the behaviors of these two species. Right? I should build that into the model. If I know that these two species are predator-prey, I shouldn't allow there to be any chance of there being two sort of predator, two positive alphas in there. Everything else here is just sort of how you fit the model. If you've heard of Metropolis Hastings or Gibbs sampling, it's a combination. The way that I fit this model is a combination of both. Okay. Okay, so now we're going to get to the application and hopefully the fun part here, which is the actual data of interest. So when I was talking about, when I introduced the talk, and I said I was working with Sarah Roberts, she's the one who actually got this data for us. So this is data from the Northeast Science Fisheries Center. And they have a lot of data, but we are focusing on bottom trawl surveys that are conducted by this center. And so what they do is they've already designed a nice sampling protocol. So they have taken this sort of this eastern side, the Atlantic Ocean bordering the continental US, and they have already predefined sort of sampling regions. So they go out, they did this before, and they kind of make different size aerial units to sort of adjust for depth level within area units. So trying to have constant depth across all of these aerial units. So that's why some are a lot smaller than others. And what they do is based off the area, they will go out and they will conduct couple of trawl surveys. They do a little tour this area, and they'll park their boat for about 20 minutes and tow a trawl, and then they'll bring it all up and they'll cache all the species that they caught their net. And they'll do that for all of these little aerial units, which I will call strata. And so they do this in both the fall and the spring, but we're going to focus on the fall surveys only, acknowledging the fact that some species do migrate seasonally between fall and spring, and that complicates the model even further. So we're just going to focus on fall surveys. And we have data from 1986 to 2017, and we're focusing specifically on the Gulf of Mexico area. And this, so there's 28 areas here. And so the data that we see, those Ys, those noisy data that we have, are the observed catch per unit efforts. So it's the kilograms of biomass per 20-minute tow for each species in that net. That's the data. Okay. And so what we can do just for results later on, even though this isn't necessarily what the species, how the species behave, what we do is we take up the Gulf of Maine area and we just sort of ad hoc make 4 regions for interpretation later on. So we decided to call the northern region the Gulf of Maine, the southern region, the middle, and the interior region. Just to see, are these species behaving differently in these different regions later on. Okay, so some of the look at the data and look at some of the covariates that we have. So this is looking at Atlantic cod, for example, was one of the species that was available. And so these are sort of time series, and in those 4 regions that we made. This is the observed data, so intro, middle, north, and south. And this is the average sort of catch per unit effort across all of those 28 straight up. So you can see already this unit is extremely noisy. right, in the inshore region for a while, until like about 2000, there were very little cod inshore, and then all of a sudden it's this huge spike in like the early 2000s. Whereas there's a little bit still noisy, a lot of stochasticity here, but a little more predictable behavior, not a smaller range of values, right? So already this data is going to be messy, and so the messier data I have, the harder it is to make inference usually. There's a lot of variation. We sort of try and keep that in the back of our mind as we fit our models. And then we have one of the really important covariates, not the only one, but one covariate that we have or predictor is the sea surface temperature within each one of these aerial units. And so what we have done here is we've averaged the sea surface temperature within sort of 6 or 7 year time windows, just for illustration purposes, to show, oh yeah, the waters are getting warmer over time, seeing how that might. So we're just acknowledging, okay, there's maybe going to be some effective temperature, and here these are just maps of the difference. So we can see that between from the 2004-2009 time window to the following 7 years, there's a huge increase in the temperatures here. So we acknowledge that there is some change in temperature, and maybe that'll have an effect on not just the growth rate, but also how these species are moving. Okay, so what we did here, I'm showing you this prior food web, which is essentially showcasing a few things. I don't actually know how many species were in the data, but there were hundreds at that magnitude, and we're not going to fit our model on all hundreds of these species. That would probably take months, years to run. So what we did, what I collaborated with Sarah on, was she was choosing really important species that were probably known for some of them to interact with each other. So she chose this community. So we pared it down to 8 species that were both of economic interest, but also from ecological as well. And so she chose Atlantic cod, Atlantic herring, mackerel, goosefish, haddock, redhead, silverhead, and spiny dogfish. And what we do as a prior, so remember those community dynamics, the effect of a species on another species, the actual magnitude of the effect is unknown, right? But we want to estimate those, but we can have some prior information. So you're having different colorings here. So what Sarah did was she got access to gut data. So some people opened up the stomachs of these fish, and we're trying to analyze what proportion of the gut was comprised of this species versus that species versus that species. So if there was at least some percentage of, let's say, herring found in the guts of silver hake, then it was determined that we a priori decided to say that herring was a prey of silver hake, because there was a lot of herring in the gut of silver hake and not the other way around. So that's where we can put in some of that expert information, right? Someone went into the stomachs and was able, and so then Sarah decided, okay, this is a predator of this species. Maybe they both eat each other, so maybe they're both positive, or they're both, we have that kind of competition. Some species were found within their own guts, right? So silver hake and cod were eating themselves. It's not perfect. Some of this gut data gets apparently, I guess I believe it, that the gut doesn't tell all. So once it gets into your stomach, it's kind of hard to tell what's what. So it's not a perfect food web. But you'll notice that there are some species that don't have arrows pointing at each other at all. So those are ones that we gave priority, said there's no direct pairwise interaction between these two, because we didn't see any of the other species, any of the one species in the gut of the other, for example. And then the width of these arrows is sort of saying, how sure are we about, how large are we allowing the range of the magnitude of those alphas to be? Because larger magnitude alphas will mean a stronger effect on the growth rate. And so for a lot of these, usually sort of bigger larger fish are going to probably have a larger impact, and so those will have wider possible ranges. But so the width is just sort of embedding that information. Another obvious issue with this food web, right, is that we're omitting other important species that weren't there. For example, like squid, we know makes up a big factor into the diets of a lot of these fish. But that was we just kind of put them all in here. Okay. Just to say, I don't want to step through all of this, but I just want to say at a high level, right? If you go back to when you're saying those journal articles that I showed at the beginning, fish are moving according to their tracking temperatures, right? There's a lot of research showing that they are tracking temperatures and a lot of them are going northward towards the colder waters. We're trying to incorporate the idea into this our redistribution that fish are moving towards some thermal optimum that we don't know what it is, but they're trying to find a place that's best for them in terms of temperature. But we don't know what that thermal optimum is. So we just put that in as a function into a function for redistribution, and we try and estimate the thermal optimum for each species. That's at a high level. So no, we're trying to acknowledge the fact that these species are moving. We're trying to build that and they're moving towards a preferred temperature window gradient, preferred temperature, and then we're going to try and estimate that temperature. That's all I'll say about this. Okay, so then the results, which is the exciting part, except these weren't all that exciting, unfortunately. So these are the posterior distributions for the environmental species interaction. So we had, this is the density independent growth rate, so how much they change irrespective of outside influence. These are the posterior distributions, so how the distributions of these coefficients after data and prior for the effective sea surface temperature interactions. Then we had two more that were North Atlantic oscillation, AMO. Yeah, that, whatever that is, I forget, I knew it at one point. Yeah, something, but they're cyclic or something, I don't exactly know, but when you collaborate, they tell you things that are clear, isn't it? And so what I've done is I've boxed out in colors, coefficients, parameters whose posterior credible intervals, 95% credible intervals excluded 0. So we're talking about significance, which some people care about. These are the ones that were significant in the base after fitting the model. And you know, and you can interpret it as you wish. So it seems like as the surface temperatures get larger, right, they're having negative effects on growth rate, which might make sense for some of these. NAO is really not found to be having a big influence, measurable influence on the growth rates. But once again, the data are really noisy, and so it's hard necessarily to pin down exactly the effects, and there might not be. But so those are posterior distributions for ESI. What we care about are the posterior distributions for those community dynamics, the species interactions, and I'll show another photo in a second, our image. But essentially, I could have said this in the previous slide, the blue line sort of represents our prior beliefs. So we were believing for the interaction between goosefish and haddocks, this could have been any, the growth rate coefficient could have been any value between -0.1 and 1. That's what the straight line is. But then in the face of data, the data is like, no, actually there's a negative effect, a pretty negative effect here. And so you can see that for some of these, there is a lot of learning going on. We're moving away from that straight horizontal line, and there's a lot of posterior learning happening. But there's also some wonky behavior going on as well. But what we do is we're saying, okay, there's evidence of dynamics for sure in here, as well as, and we're learning about the magnitude. And so when we compare sort of posterior and prior, I'm omitting some of the arrows that weren't showing a lot of change that were from the prior. But you can sort of see pared down what are the most important from our model interactions going on. And then what Sarah did was she was trying to interpret these in terms of a policy perspective. Are we seeing more negative effects than the positive effects that are showing in the model? What does that mean for ecosystem management? What are the species that might be having a big influence on how the community behaves? These are estimates of the posterior mean estimates for those thermal optimum. So trying to say, oh, we're trying to estimate where the species are going. And so these are the different species in our community. And these are 1 degree Celsius windows, and sort of the proportion of biomass, latent true biomass for each one of these in our model, along with the estimate of that thermal optimum. So I'm sorting it in terms of thermal optimum here. So based on our model, the herring and spiny dogfish and cod are preferring colder waters as that thermal optimum compared to the fish over here. But also, where are all the biomass happening? As it turns out, there was one year where there were just a ton of herring found in a very warm place. So you'll see this bright yellow up here. We're just trying to understand, we have this thermal optimum. Are the species abundances really aligning with the optimum? And if not, that might be a signal that they're not happy and they're moving other places. Maps. People like maps. Trying to estimate that latent log biomass over time for Atlantic cod. But what we really were caring about is trying to understand how are the trends changing over time. So what we did over here essentially, we took those estimated true abundances, the log version, and we just fit a regression, a very simple linear regression for time to see what are the trends over time. And then these are the colorings for that coefficient for year. And then all this is really saying, which maybe you can see from if you were looking at the raw data, but as time goes on, there's a lot fewer cod living down here. And there's also fewer living, there are fewer, the abundances are decreased, you could say, in these regions, but especially in the southern Gulf. They're really moving out of there over time. I know I'm almost done. So what we were trying to do is we built this big model to try and embed, right, different biological mechanisms in our model into in a coherent way. But the big question is, okay, you did all of this. Can you actually separate any of the effects of the growth and the redistribution? Like you did all of this, but were they just like were they just oscillating back and forth? It was like when they grow, is it like, oh, you see an increase in growth, but then the redistribution has caused it to decrease, so they're balancing each other out. And so one of our reviewers really wanted to see this. And so what we have here is the estimated annual changes in Atlantic cod biomass due to these two biological processes, growth and redistribution for different strata. So these are just numbering strata along with error bars. And so we're trying to see, is there just a pattern where we saw an increase in growth, redistribution is going down, so they're canceling each other out, essentially? Or if not, can we say how much of the change in abundance was due to growth versus redistribution? And so that's what we're looking at here. So for example, in this region, there's a lot more pink that are up here, so that a lot of the increases in abundance were due to redistribution as opposed to due to growth. For example, whereas other places like the black here is really dominating, so growth is really dominating the changes in abundance of cod in this region. Okay, so in summary, because I know I'm basically out of time, what we try to do is to develop this big model that brings in some biological processes. Hopefully I've convinced you that we did our best. And maybe you can do better. I think it's always about, we don't assume that this is the best model out there, but it's one step forward in trying to combine ecology with just a big statistical model. There are many limitations, as our reviewers were extremely happy to point out. For example, there's immigration into and immigration out of the Gulf of Maine, and we said, nope, these fish are stuck in here. They cannot leave, which is obviously not true, but that could be like one risk that you add to your model. And we also only looked at a subset of the community, so we only looked at 8 of the species, but there are definitely more, especially fish having birds have a large predation effect. And there's also a lot of other important environmental predictors that we are missing that we just did not have access to, for example. And that's it. Thank you so much.
Thank you so much. I want to ask Jenny and Sebastian. I think you guys are in Mark by Baby and get to ask the first question.
Hi, I'm really interested in what you mentioned, and I'm also currently working on a long-term biological molecular program. So I was just wondering how you deal with those environmental factors like water quality data, the pH or water temperature, those kind of stuff, or like how you choose them to put into your model.
So which predictors to include? Well, as the statistician, I let my collaborators tell me what to include. But no, I think a lot of it is, there are a couple of answers to that, right? It depends on what stage of the modeling process you're on, right? Some people are extremely interested, they don't know if certain predictors are going to have an effect. And so they fit a model and they run hypothesis tests on like the significance of coefficients or something. And just to say, I want to learn which ones might be affecting the outcome. Other times, it's just you read the literature, you read what people have found in the past. People have spent decades researching, and so they know, okay, pH definitely has an important effect. So let me throw it into the model, and so that's where a lot of that lit review comes in. And so it's sort of, yeah, I think it's a lot of understanding your system, which comes with time. It comes with your advisor, I think. And I don't know if that's a satisfying answer, But ask your advisors, I would say, or ask your peers, right? Look into the papers, and see. So, right, and sometimes it's also not just about what you want to have, but what you have access to, and is it measurable at the scale at which you're observing your data? So our data were observed at the aerial unit scale at a stratum level, but maybe certain processes occur at an even finer grained level than that. Like there's an I don't have blanking right now, but you might have different scales of interest. And so maybe even though you know that this covariate should have an effect, it might not be popping in, it might not be showing up as significant in your model because of the different scales of interest. It's called, yeah.
I don't know if Sebastian has a question.
I have two questions. So first, very interesting sharing and very huge project. Yeah. So I have two questions. For the first one is, I want, do you have explanation to the decadal process like the North Atlantic oscillation and AMO Atlantic Meridian oscillation to the distribution? I want
The explanation for like why we include like the NAO and AMO in our model.
So the result.
The result.
Yes.
So I don't actually know because as you can tell by me not knowing what those acronyms stood for. I didn't have a great interpretation for it, so that's what I leave the ecologists in the room to interpret for me. But I think a lot of it was, why did they include it to begin with? Sarah had seen papers where they included NAO and AMO, and they found significant results. But I think in those papers, the regions of interest were much larger, so they were probably seeing larger significant results in there, whereas in the Gulf of Maine, it's a relatively small region considering the whole Atlantic. And so maybe there was just not that much variation across time within those regions. So I don't have a great, like, why are these negative? Like, what is the interpretation here? But I'm sure that your advisors do. Or there are other papers out there that try to shed light on that. So, yeah.
Can I ask about
Yeah.
this? So about the growth part, not just like, how do you add the random part into?
Yeah. So where does that random stochasticity come into play? So, right, so we had this big, I'll go to this nicer one. We said in this two species case, we have this deterministic equation, right? That has growth is determined by density independent, density dependent, and environmental species interactions. And so I built those in all into one, and we said, okay, abundance at time t plus 1 was exactly equal to the growth. This is essentially where you were before, plus the effects of growth. And so in our model, we get rid of that equation, and we put a normal distribution. So what does this look like? Remember, these W's are not known to us, right? These are latent true abundances that are getting informed by priors for other things, but also the observed data, the Y's. And so in our model, we put this distribution, we're saying the distribution of these W's is normal according to this. And so when we what we actually need to do in the model is we need to sample these W's, we need to estimate these values. And the way that we estimate them is by saying we believe that they are normally distributed. So we're drawing them from, as it turns out, a normal distribution with error involved into it. So what we say is we're going to have this big normal distribution centered at M. And this is not exactly how it works. But you can imagine if I were just to draw from this, it's a normal distribution centered at M. But according to I'm not going to take a value M exactly. According to some error, I might sample close to M or further away, depending on the amount of error in my model. And so this error gets propagated through, but essentially it's saying, instead of saying Wij t plus 1 is exactly equal to this, I'm sampling with randomness from another distribution. And the randomness gets built in with that t plus 1. I don't know if that makes sense, and I don't know how.
It's very interesting. Have you tried? I'm worried about if this W, if it will go so far that it will not that big to the normal distribution.
Sure, yeah, that's a good question, like, that's a great point, like, are these W's truly going to be normally, like, is this the right assumption to make, that these W's are normally distributed? Great question. And that depends on the system of interest, right? So for us, we just put this normal, but maybe you have some system where actually the latent true abundances are going crazy. But this is about, how, what can I say? This is how far they can go beyond M. It's sort of governed by this error term. So if sigma squared is really large, you're allowing for very large values to move away. And if you don't believe that they should be that different, then you should have a prior that constrains that error term to be quite small. So you can put prior information to say, I don't think that the growth should change that. Like, I think it should behave according to this equation, and pretty much according to this equation. I'm going to set my prior sigma squared such that the variance has to be very close to 1 or 2 or something, depending on the scale of your data. Whereas if you're really not sure, and you're like, oh, these populations are crazy, they're extremely erratic from year to year, I'm going to have a prior that allows sigma squared to be really large. And therefore giving more flexibility, more room for these W's to take on more values.
Yes.
So great questions. Yeah, support.
Great lecture. It was really interesting. I'm not from the marine science.
Me neither.
Yeah. Well, this is cool. First, when you're coding this, are you using R?
I am using
Yeah,
R.
awesome. Okay. So when you code, and this is the slide I actually had a question about, so this is great. When you fit it into discrete time steps and you have it into like at the end where it's like that MIJT form, are you still in your code individualizing like your density, your alphas? Like, are they all individual variables, or is it like an overall assumption?
So all of the alphas are individual parameters.
Right.
We update, but we put them into one big matrix actually, and then we update them according in a matrix form. So they are individual parameters, but when we learn about them, we actually do a lot under the hood to make the inference a lot faster. So we put it in a really weird form. So you could treat them, in your code, you could have lines separately for each one of these alphas. We end up saying that's annoying because you don't know how many you have before. We want our data to be, we want our code to be as reproducible as possible. So we put them into a very specific kind of matrix form that allows just one line to update all of them.
Okay.
But that's not always necessarily going to be able to do that.
And so when you use your transport matrix or H matrix, and then against like it's against the W, right? Okay. And so then you have another matrix within your W that you're
That's right.
looking at. Okay, so then it's just like
It's matrix multiplication.
right. Okay.
Yeah.
And is that matrix the same size as the sensitivity matrix? Like the N, like or is it different? The alpha matrix is that
They're different sizes.
They're different sizes.
Yeah, because the redistribution matrix says, what is the chance of going from these area units to these area units, right? Whereas these alphas, there's not as many alphas as there are area units, right? These are the number that we have is going to be at most capital J squared, right? Whereas in our redistribution matrix, it's going to be n squared values, because you need to say for each location, where can I go? So they're definitely different sizes.
I have one more question. And then something else. For the Bayesian modeling question, you said about how like one step, is it independent of itself, or is it just based off a previous step and that's it?
It depends on the actual problem at hand. So
Right.
I'm kind of like glazed over a little bit. But if you look at this equation right here, this model, I probably should have had a little bit more here. I'm kind of omitting some important things, but if we look at this deterministic equation, right, we're saying growth at time t+1 depends on is equal. Ignore the stochastics.
Yeah.
It's exactly equal to this. And you'll notice there's only the Wijts. There's no Wij -1. There's no Wij -2, that kind of thing. And so we will say that once I have these Wijts, the growth at time t plus the abundance at time t+1 is independent of anything that came before here.
Okay.
That's
so
what I mean by
on T. If it's
That's right.
T+1.
And that's, exactly.
So then if it's T+2, it's dependent on T+1.
That's right. In our model, that's an assumption that we have.
Okay.
Right, but you could have different assumptions. So maybe you have a model. You could have a model that says actually I think it depends on the past 5 years. Like I think like maybe it's some kind of lag effect in the past 5 years.
Is that not a Bayesian?
No, it still would, but the Bayesian model allows us to break it. So Bayesian is not about necessarily that convenience, the t being independent of t -1. But when you make assumptions like that, the Bayesian model lets us break down the inferential component into smaller building blocks. Which is, if you ever get into Bayesian, would help you, maybe it would make a little more sense, but essentially it's saying, I have all these parameters. I have to estimate all these W's, I have to estimate all these rows and all these alphas. Rather than estimating all of them at one big time, let me estimate the alphas first based on what's important to learning about the alphas. Then let me estimate the rows depending on what's important for the rows. Then let me estimate the W's by what's important for the W's, but then I can even break it down further by saying the first W's depend on just nothing, and then the W2s are gonna depend on the W1s, et cetera. So it's about breaking it down as much as we can.
We're a little, well, Stephen, why don't you end the.
Oh, no.
Go for it.
So much pressure, because I've got about 5 different questions I can ask, I've got to figure out which one to ask now, okay. I think if I were to point out one thing that was really interesting about your results is this reduction of the community matrix. Because that's actually really useful, if you actually can reduce the work interactions. Then actually you get a little bit of focus on the things that matter in terms of management and stuff like that. The question I wonder about is how dependent do you think that the actual nature of that reduction is on things like relative abundance of the species, the ability to detect
Yes.
those interactions?
Sure. It's probably quite sensitive, and I bet if you added more species into the community, things might change as well. So I do think there is, like, you can't just take this as, like, this is a great caution, right? You can't just take these results and say, this is exactly how these behave, let's do benefit policy according to that. So we performed a little bit of sensitivity analysis, and it's kind of in our paper, trying to estimate, not so much about the community, like recovery, so that was under the assumption of true alphas and saying if we can predict abundances for another location. Certainly the quality of your data will influence the kind of inference that you get, and so when it comes to non-detection, I think that's a huge issue, right? So a lot of these species might have been, were actually observed, not in this data actually, in another data set, were observed zero abundance, and that really just makes it very difficult to estimate these relationships. And so most likely quite sensitive, honestly, I would say so. So when we look at that posterior food web, we're trying to say, okay, can we pare down this, what we had, our beliefs from before, is it changing at all? I guess that should be useful for helping guide management, but not sort of as the end all be all, right? Like we are seeing, these are the strong effects that are coming in under these assumptions, right? Statistics is all about assumptions. So under these assumptions, these are the effects that we're seeing. These are the interactions that we're seeing. What should we do with that information? Knowing that this might not be exactly the way that they do. So maybe not a satisfying answer, but surely anytime you're going to change, like the quality of the data, of course, is going to change the how good.
Well, thank you so much.